Hello, my name is Michael Stickles, and today I'll be presenting my thesis project for the University Honors Program titled Investigating BRCA2's Role in Crossover Regulation in Mouse Spermatocytes. So what is BRCA2 exactly? BRCA2, or breast cancer-associated gene 2, is a tumor suppressor protein found nearly universally across the three kingdoms of life in some form. This particular gene has become a rather hot topic in recent medical and scientific literature due to its intimate relationship with hereditary breast cancers. This graph here depicts the staggeringly higher risk of developing breast cancer for carriers of a mutated BRCA2 gene as opposed to relatively low risk for non-carriers. While many of the functions of BRCA2 are still being investigated, one of its main roles is the regulating an error-free mode of DNA repair, known as homologous recombination. Double-strand breaks in cell DNA can be caused by intrinsic or extrinsic factors. To repair this damage, BRCA2 enables the loading of RAD51, as depicted by the diagram, to facilitate homology search, and thus repair the DNA. The role of BRCA2 in regulating homologous recombination has been well studied in somatic cells, but the role of BRCA2 in germ cells is still largely unknown. This research project was designed to investigate how exactly BRCA2 influences crossover formation and regulation in germ cells, or in other words, cells going through meiosis. So what is meiosis exactly? Meiosis, simply put, is the process in which cells divide to create haploid gametes, or sperm and eggs. One of the main ideas I'd like you to walk away from the slide with is the fact that meiosis is distinct from other types of cell divisions. Here, daughter cells end up with half of the number of chromosomes in comparison to the parent cell, as you see here depicted by the diagram, meaning this is a type of reductional division. One gamete receives one copy of DNA from the parent, and the other gamete gets a copy from the other parent's DNA, and they unite to form a diploid organism, which eventually goes on to develop into the mature organism. So what occurs during this reductional division we call meiosis? Crossover formation between chromosomes of the same structure is the first step to reductional division. Crossovers also lead to an exchange of genetic information between the two chromosomes. If you might have guessed, this process must be tightly regulated to ensure genetic information is not altered nor damaged. That being said, cells follow strict rules for crossover formation. One of them is that each chromosome must at least get one crossover. And if there is more than one crossover per chromosome, then they must be far enough apart to ensure that there is structural tension minimized between the 2 points. So, provided BRCA2's role in regulating homologous recombination, I sought to understand BRCA2's role in crossover formation and regulation during mammalian meiosis using mouse models. However, traditional knockout mutants could not be implemented in this experiment due to the inherent embryonic lethality presented by the BRCA2 knockout mouse model. Looking at the diagram, developmental defects can be clearly visible in the mutant embryo, which result in inviability. So we instead used a conditional mutants, which made use of a transgene called Cre recombinase to mutate the BRCA2 gene. This allowed for mice spermatocytes to have the same BRCA2 genotype as traditional knockout mutants, but without the embryonic lethality. Let's take a look at how this process works. So, to begin, each somatic cell in a developing mouse has one copy of the floxed BRCA2 allele. When I say floxed, I'm referring specifically to the LOXP sites around exon 11 of the gene, denoted by the red triangles. These LOXP sites were artificially engineered into the genome to help our transgene facilitate the mutation process, which will be covered later. Upon transcription and translation of the floxed BRCA2 gene, a functional BRCA2 protein will be made like so. Now, let's take a look inside the spermatocytes, other known as the germ cells. In addition to the floxed BRCA2 gene, these cells have a germline-specific gene, meaning it is not found in somatic cells. This gene is called STRA8, or stimulated by retinoic acid 8. Upon transcription and translation in spermatocytes, a functional BRCA2 protein and STRA8 protein are created because they are both present in the cell. This cell is known as Cre negative because there is no Cre enzyme in there yet. However, to induce our conditional mutation, we inserted a transgene under the control of the STRA8 promoter. Now, this enzyme will also be expressed upon meiosis in a similar pattern to straw 8. This cell is now called Cre positive because it has a Cre recombinase transgene in it. Now, this protein subsequently recognizes the LOXP sites on the floxed BRCA2 allele and removes exon 11 from the gene like so. Now, the BRCA2 gene is permanently altered and is considered mutant. It can no longer produce operational copies of the BRCA2 protein, only mutant copies that do not perform properly. Now, the mouse can develop to adulthood, avoiding the embryonic lethality thanks to the timed natural expression of the STRA8 promoter. Now that we have a solid background for my experiment, let's jump into the process. To harvest the data was a lengthy task. First, we had to breed mice with the desired phenotype. As discussed previously, we need a control group and a mutant group. Mating pairs of mice were set up optimally to yield pups with our desired genotypes, as depicted by the diagram. Next, we had to verify the offspring genotypes. Using PCR, or polymerase chain reactions, the genotypes of the litters from the arranged mating pairs were determined to choose viable candidates for further research. Next, we spread out the nucleus on microscope slides to be further stained and visualized. Nuclei from the spreads were stained using a process called indirect immunofluorescence. Indirect immunofluorescence is a technique used to visualize the proteins of interest. In this process, the proteins of interest were on the chromosomes were stained with an antibody that binds to a particular region of the protein called an epitope. The primary antibody only can recognize and bind to that specific epitope it was designed for, meaning this process is very specific. Notice here it will not fit onto the other protein and only onto the protein of interest. After this was complete, a second wave of staining was done with secondary antibodies. The addition of differently colored fluorescent tags allows us to visualize the protein. Once it binds on to the first antibody, we can visualize the protein of interest easily. The results look something like this. You can see the differently colored proteins here. From these images, we can gather our data to provide us insight on how BRCA2 influences crossover formation by the amount of crossover foci per nucleus. These values were then integrated into a spreadsheet and analyzed for computational software called GraphPad Prism 8. So here is our first set of data. The center box shows mouse spermatocytes from control and BRCA2 mutant mice, with the 20 paired chromosomes stained in gray, centromeres stained in blue, and the sites of crossovers stained in red, marked by a protein known as MLH1. If you look in this diagram here, the protein MLH1 is denoted by the flashing region. On the right, over here in the graph, the number of MLH1 foci per nucleus is plotted for the control and mutant groups. We did not observe any significant difference between the control and BRCA2 mutant spermatocytes, but there was a higher variance in the mutant group, as you can see from the wider distribution of data points. This could suggest that crossover formation was less precise in the mutant group. Although crossover formation of BRCA2 mutant spermatocytes looks largely normal, we next sought to investigate if crossover regulation follows the normal pattern as well. To understand crossover regulation, we used Velocity software to analyze the nuclear spread images, where I painstakingly measured the interfocal distances and synaptonymal complex lengths like so, using small rectangles connected to give us a length value. These values were then integrated into a spreadsheet and then GraphPad Prism 8 like the previous data. Here is our second set of data. Looking to the left, plotted are graphs of the synaptonymal complexes, or chromosome lengths, in micrometers of the control and mutant groups. The synaptonymal complex length is denoted by the graphic to the left of the axis. Notice it spans from the centromere all the way to the end of the chromosome. There is no significant difference between the two groups. Looking to the right, plotted are the average interfocal distances in micrometers of the chromosomes for the control group and the mutant groups. The interfocal distance is demonstrated by the graphic next to the y-axis right here. And again, we did not observe any significant difference between the two groups. In conclusion, there is no significant difference between the number of crossovers per nuclei, interfocal distance, or chromosomal axis length between the mutant group and the control group. Given BRCA2's role in homologous recombination, these results are quite surprising. The data thus far did not give us any significant evidence for BRCA2's role in crossover regulation or formation. However, as good scientists do, we need to contrive some possible explanations to explain the lack of difference between the mutant and control groups. One option that we can simply conclude is that BRCA2 has no role in meiotic crossover formation or regulation at all. Therefore, the removal of BRCA2 would not change the length of the chromosome axes, the MLH1 counts, or the average interfocal distance. However, this hypothesis is hard to believe, though, as we all know that most cells in this mutant are not even able to initiate any homologous recombination whatsoever, as shown by this nucleus with 40 unpaired chromosomes. Or, additionally, we may hypothesize that somewhere in the process, an error occurred. It is known that Cre recombinase excisions can be leaky, meaning they're not 100% efficient. This may have accounted for some totally normal crossover formation in BRCA2 mutants. As shown here, wild-type spermatocytes progress normally through meiosis, but spermatocytes in mutant BRCA2 show high mortality rate, up to 80%, as reflected by significantly reduced testes size in BRCA2 mutants. I've inflated the graph so it's easier to visualize. Most cells do not survive beyond the zygotine stage. So we speculate that cells that do make it past the zygotine stage actually possess the wild-type BRCA2 because of the leaky Cre recombinase instead of the mutant BRCA2. This could be why no defects were detected in crossover regulation. Now we are also investigating a similar study using another Cre line under a different meiotic promoter called SPO11, which expresses in postnatal day 11. Hopefully, this can provide us the insight we were looking for on BRCA2's role in crossover regulation. I'd like to acknowledge the people that helped me contribute to my project, namely Dr. Hunter, my PI, Dr. Sumit Sandhu, my mentor and supervisor, Hunter Laboratory, and all my lovely lab mates. This concludes my presentation. Thanks for tuning in, and I hope you learned something.